Hello, today I'm going to present to you the materials that you need to prepare for this lab. This lab is about ocean acidification and will allow your students to do experiments regarding the biodiversity in our world's oceans. From the tiny plankton that we can't even see with our own eyes, to the mammoth yet magnificent blue whale that dwarfs even the largest and mightiest dinosaurs, are all threatened by the effects of ocean acidification. Let's begin with the materials that you'll need to prepare for this lab. There are 2 stages in this lab, and the materials that are required for each lab include, for the Bubbles Protocol kit, one 500 milliliter beaker of 100 milliliters of prepared salt water or seawater, one 500 milliliter beaker of 100 milliliters of pH tap water or 7 bottles of water, one straw, 1.5 milliliter tube with 1 milliliter universal indicator, one disposable transfer pipette, one sheet of parafilm, and that is all. For the Shells Protocol kit, you should add these items: one 500 milliliter beaker with 150 milliliters of white vinegar, 2 strips of universal pH paper, 1 pH color chart, a set of weights or textbooks, 1 Sharpie marker for writing on the shells, 1 bag of mussel shells. For each bag, they should contain 2 untreated shell halves and 2 pre-treated shell halves. You can tell by L means low exposure, which is 4 to 6 hours, and H means high exposure, 16 to 18 hours. You'll also need 2 balances and 1 tub of ice for the whole class. A timer is needed for both stages of this lab. Also, print out the protocols so that you and your students know what you're going to do in this lab. For this lab, you'll need to prepare seawater. If you live near the coast, obtaining seawater would be ideal. If you don't, aquarium salt is provided in the kits so that you can create your own salt water. Remember to prepare each of the concentrations of water for the protocols differently. For the Bubbles Protocol, make sure your tap water is pH 7 and mix each tube labeled Bubbles with 1 liter of tap water. For the Shells Protocol, mix the tube labeled Shells with 1.5 liters of tap water. If you get seawater, you should refrigerate it for the best quality. Remember to separate and label the 2 seawater concentrations for the 2 labs, the Shells Protocol and the Bubbles Protocol. Prior to starting this experiment, have all the materials be ready. Remember that the items such as vinegar need to be purchased, since they don't come with the kit. Before the lab begins, remember to assign the students active roles in the project and split them up into separate groups. Then distribute the materials to several stations for the students to become more actively engaged in the project. On the day of the Bubbles Protocol part of the experiment, have the ice and the heat source be ready for the students to use in order to test how the rate of reaction changes under experimental conditions. Proper lab prep will help your students understand these complex concepts as they become more actively engaged. Good luck on teaching the effects of ocean acidification to your class.